The key link is really the fact that are the major producer of adiponectin, that adiponectin with obesity, with diabetes, and it's a recently discovered molecule, barely 5-6 years ago. So it came late in the genome discovery, and now it's catching a lot of attention and is clearly linked with obesity, and now for the first time with cancer. Adiponectin is a hormone that is secreted by the fat cells in the body, and a lot of research has gone into adiponectin in the past few years, and adiponectin has been found to be associated with risk for cardiac disease, risk for diabetes, as well as with various syndrome linked with obesity and cardiac disease. A recent study showed that the level of adiponectin in the blood was associated with colorectal cancer. We picked some variants in the adiponectin gene and the adiponectin receptor that had been already previously studied in other diseases like cardiac disease, diabetes, etc. And then we chose these SNPs, and then we tested them in our patients with colorectal cancer and in the control. And that's where we found that some of these variants were indeed associated with colon cancer risk. And then when we replicated this finding, one of the 4 that we found in the first study panned out the same way as in the first study, which provides strong support for the notion that these variants is associated with colorectal cancer risk, and tells us that probably the region of this adiponectin gene that carries this variant has some function, carries some function in terms of risk for colorectal cancer, but more work needs to be done. So based on previously demonstrated association between the adiponectin level in the blood and risk of colorectal cancer, we thought that variants, genetic variants of the gene for adiponectin and its receptor might be associated with colorectal cancer. And to test this hypothesis, we recruited patients with colorectal cancer as well as controls in a first study of about 1,100 patients and controls. And we chose 10 variants of these 2 genes, so 5 for each gene, and we determined the frequency of each variant in each group. And we found that 4 variants, 3 in the adiponectin gene and 1 in the adiponectin receptor, were associated with colorectal cancer risk in the first study, which means that the frequency of these variants was significantly different between cases and control. Like all studies, these needed to be validated, and that's what we did with the second study, where we took 199 cases, a patient with colorectal cancer, and 199 control that were very well matched. Each patient with colorectal cancer was matched with a healthy control of the same age, gender, and ethnic status and location. They all were from Chicago. And we determined the frequency again of the same 10 genetic variants among patients with colon cancer and among control. And we found that one of the 4 SNPs, or 4 genetic variants, which had been found to be associated with colorectal cancer's risk in the first study, was also associated in the second study to the same degree. So that provides strong support for the novel notion that a variant of this gene, the adiponectin gene, may predispose to colorectal cancer. We did find an association that was consistent in the 2 studies. The association suggests that given variant of the adiponectin gene is associated with a 30% increased or decreased risk of colorectal cancer, depending on which group you look at, and thereby indeed impacting the risk for colorectal cancer in a significant fashion. Whether this will pan out ultimately will depend on the validation of these results in other studies by other groups, and this may ultimately lead to the identification of a blood test that would help to identify who is at risk of developing colorectal cancer based on these adiponectin gene variants. Colorectal cancer is a very common disease. It is the second cause of cancer death in the US among non-smoker and the third most common cause of cancer deaths among smokers. This disease is very common, and we know that about 1/3 of colorectal cancer cases have at least one family member, first, second, or third degree relative with the same disease. So we know that this disease is inherited in approximately 1/3 of cases. However, our knowledge of genetics only accounts for maybe 5 to 6% of colorectal cancer, which means that an additional 20 to 25% of inherited colorectal cancer cases are inherited due to factors we don't know yet. And that's why the search for a novel genetic marker of colorectal cancer is important. It is important for several reasons. Colon cancer, in contrast to many other cancers, can be diagnosed at an early stage, and not only diagnosed but effectively treated at an early stage through colonoscopy. However, the current guidelines recommend colonoscopy to be done for the general population at the age of 50. By the age of 50, some patients have already developed colorectal cancer and thereby were not offered screening that may have helped them. And we know also that maybe screening by the age of 50, even though it's a good thing to do, screens a lot of patients that may not need screening at that age. And the fact of the matter is that only 40% of the population that should have screening has screening for colorectal cancer. These markers are not so much randomly, but are chosen based on the fact that they represent an entire region of a gene. However, by doing that, they may miss some region within this gene that are indeed more potent in terms of their impact on the risk. So indeed, the next step will be to, after these findings are validated, to determine which marker within this region are indeed the best markers for colorectal cancer risk. And that's what we intend to do in future studies, is to narrow down the region that is associated with colorectal cancer risk by performing several studies that will include resequencing of the entire region to identify which region are truly the most significantly associated with colorectal cancer risk. Whenever you discover a gene involved in increased or decreased colorectal cancer risk, it may open some new avenues for therapeutic intervention. And while this is probably still not in the immediate future, it definitely could lead to the discovery of novel therapeutic modalities that will make use of these adiponectin gene and its related pathways. We have many good drugs to treat colorectal cancer, and I think it's important to remember that barely 10 years ago, a patient with metastatic colorectal cancer had very little chance to survive over 1 year. And today, with our current drugs, the average life expectancy for patients with metastatic colorectal cancer, which cannot be resected surgically, exceeds 2 years. So there have been more than a doubling of the survival of patients with colorectal cancer, and drugs have become very effective at doing that. However, most patients with metastatic colorectal cancer will ultimately die of the disease, and there is certainly a lot of room for improvement, and novel therapeutic agent that could be added to the current drugs that we use would be very much welcome.